You are looking at a simple $1 plastic jug resting on a windowsill. Inside this container, something is alive, bubbling and breathing, quietly exhaling into the room. Remarkably, the plant positioned just 12 inches away is growing at nearly double the speed of its identical twin located on the opposite side of the space. There is no special fertilizer involved, no expensive grow lights, and no high-priced supplements. It is just a jug, some sugar, a single packet of baker's yeast, and a short length of aquarium tubing. That is the entire setup. Now here is the question that no one in the massive gardening industry wants you to ever ask. What if the primary reason your plants are growing slowly has absolutely nothing to do with your soil quality, your watering schedule, or your light levels? What if it is actually caused by the invisible gas that your plant is literally starving for at this very moment? Most gardeners spend hundreds of dollars trying to solve their problems underground. They invest in complex nitrogen blends, phosphorus boosters, and pH testing kits. They never once think to look up. They never consider the air itself. This specific blind spot is exactly what a $47 billion industry has been quietly profiting from for the last half-century. What I'm about to show you is not a simple gardening trick. It is a suppressed piece of fundamental plant biology that commercial greenhouses have been utilizing since the 1970s, charging you indirectly for it ever since. Stay with me, because the science behind this will fundamentally change the way you perceive every plant you have ever grown. Let's discuss who benefits from you remaining unaware of this. The global controlled environment agriculture market, the industry that sells CO2 enrichment systems to commercial greenhouses, was valued at $4.8 billion in 2023, according to Grandview Research. That figure is projected to surpass $9.7 billion by 2030. At the very center of that market is one simple product: pressurized CO2 gas sold in industrial canisters and piped directly into professional growing facilities to accelerate plant growth. A single CO2 enrichment setup for a mid-sized commercial greenhouse can cost anywhere from $1,500 to $8,000 up front, plus recurring canister refills that run between $200 and $600 every month. The science explaining why CO2 accelerates plant growth is not new, nor is it controversial. It has been published in peer-reviewed journals since the 1960s. Even the USDA documented yield increases of 20% to 30% in CO2-enriched growing environments as far back as 1979. This is not hidden knowledge in an academic sense. It is hidden in a practical sense. It is kept away from the home grower, the balcony gardener, and the small-scale farmer who will never be sold a $6,000 canister system. Therefore, in the eyes of the industry, these people simply do not need to know. Here's the number that should make you stop and think. A 1-month supply of baker's yeast and sugar to run a homemade CO2 reactor costs approximately $2. A comparable output from a commercial CO2 canister system costs between $200 and $400 for the same period. That represents a 10,000% markup on a biological process your great-grandmother could have figured out in a single afternoon. The $4.8 billion industry does not just sell you CO2, it sells you the belief that CO2 enrichment is too complex, too industrial, and too professional for someone like you to attempt at home. That belief is the most expensive thing you have ever bought. The story of this discovery is older and stranger than most people expect. The relationship between plants and CO2 was not discovered by a corporation. It was discovered by a French chemist named Jean Senebier in 1782. He demonstrated through careful experimentation that plants absorb what he called fixed air, which we now know as carbon dioxide, and use it as the raw material to build their own tissue. That was over 240 years ago. The foundational science of plant carbon fixation is actually older than the United States Constitution. By the late 1800s, European botanists were already conducting greenhouse experiments with elevated CO2 concentrations, documenting accelerated growth in tomatoes, cucumbers, and leafy greens. These findings were published in German and Dutch agricultural journals throughout the 1890s and early 1900s. They were not secrets. They were simply never commercialized in a way that served the small grower because there was no profit in simplicity. The real suppression happened quietly in the 1970s. As the commercial greenhouse industry in the Netherlands and Japan scaled aggressively, growers discovered that maintaining CO2 concentrations at 1,000 to 1,500 parts per million, roughly 3 to 4 times the natural atmospheric level of 400 parts per million, produced yield increases of 25% to 40% in fast-growing crops. The Dutch horticultural industry, which by 1980 controlled nearly 65% of Europe's greenhouse vegetable exports, according to Wageningen University records, built this CO2 enrichment directly into its industrial infrastructure. It became a professional secret, hiding in plain sight, documented in trade publications but invisible to the general public. What never made it into mainstream gardening culture was the biological alternative, the fermentation method. The fact is that a living organism, yeast, produces the exact same CO2 molecule that those $8,000 industrial systems generate for almost nothing, using nothing more than sugar and warm water. That gap between what professionals knew and what you were told was not an accident. To understand why this works, you need to forget everything the gardening industry taught you about feeding plants and start from a single fact that most fertilizer commercials will never mention. Approximately 96% of a plant's total dry mass does not come from the soil. It comes from the air. When you look at a tomato plant, the stem, the leaves, the fruit, and every cell of its physical structure, the carbon backbone of all of it was pulled directly out of the atmosphere as carbon dioxide gas. The soil provides minerals and water provides hydrogen and oxygen. But the carbon, the literal building material of life, comes from CO2 floating invisibly around your plant right now. This process is called carbon fixation, and it happens inside every leaf through a biochemical sequence called the Calvin cycle, identified by Melvin Calvin at UC Berkeley in 1950. This work earned him the Nobel Prize in Chemistry in 1961. In simple terms, the plant captures CO2 molecules through tiny pores in its leaves called stomata, combines that carbon with water molecules using energy from sunlight, and converts the whole assembly into glucose. This sugar fuels every biological process the plant performs, from root expansion to fruit development. No CO2 input means no glucose output. It is that direct and non-negotiable. Now here's where your garden is silently failing you. The average CO2 concentration in open outdoor air is approximately 420 parts per million as of 2024. According to NOAA's Global Monitoring Laboratory, that number sounds adequate until you consider what happens in a closed or semi-closed space. A plant growing in a corner alcove, against a wall, inside a grow tent, or in a room with poor air circulation will consume the CO2 in its immediate environment faster than passive air movement can replenish it. Studies published in the Journal of Experimental Botany have shown that CO2 levels in poorly ventilated growing spaces can drop as low as 200 parts per million during peak daylight hours, the exact window when your plant is most aggressively trying to photosynthesize. At 200 parts per million, photosynthetic efficiency drops by an estimated 50% compared to performance at 800 parts per million. Your plant is not sick or nutrient deficient. It is suffocating on an invisible level. No amount of fertilizer you push through the roots will compensate for the carbon it cannot pull from the air above it. The research is unambiguous on what happens when you correct this. A 2004 meta-analysis published in the journal New Phytologist, reviewing 159 separate studies on CO2 enrichment across multiple crop species, found that elevating CO2 concentration to between 700 and 1,000 parts per million produced an average biomass increase of 17% in woody plants and up to 28% in fast-growing vegetable crops. A 2017 study from Wageningen University, specifically examining small-scale CO2 supplementation in tomato cultivation, reported fruit yield increases of 23% with no additional inputs of water, fertilizer, or light. That is 23% more food from the same plant, the same pot, and the same soil, just by changing the air directly above the canopy. This is the variable your plant has been begging you to address, and the one the $4.8 billion enrichment industry has zero interest in making cheap or accessible for you. But biology does not care about the market, and that biology is exactly what a packet of baker's yeast is about to exploit for you. Here is where the chemistry becomes almost poetic. Baker's yeast is the same organism sold in every supermarket on Earth for less than 50 cents a packet. This same living creature that humans have been cultivating for at least 5,000 years, according to archaeological evidence from ancient Egyptian brewing sites, produces carbon dioxide as a direct byproduct of its metabolism. When yeast consumes sugar in the presence of warm water, it performs anaerobic fermentation, breaking each glucose molecule down into two molecules of ethanol and two molecules of CO2 gas. The reaction is continuous and self-sustaining for as long as sugar remains available, producing no toxic byproducts or residue that your plant cannot handle. The CO2 molecule that emerges from a yeast cell fermenting sugar in your plastic jug is chemically identical, atom for atom, bond for bond, to the CO2 molecule produced by a $6,000 industrial canister system. There is no premium version of this gas. There is no agricultural grade CO2 that performs better than biological grade CO2. It is the same molecule, and your plant has no mechanism to tell the difference. The delivery system is where physics becomes your ally. CO2 has a molecular weight of approximately 44 grams per mole, compared to 29 grams per mole for average atmospheric air. This means CO2 is roughly 1.5 times heavier than the air around it. When gas exits the tube positioned above your plant canopy, it does not disperse randomly upwards and away like a lighter gas would. It sinks. It flows downwards through the leaf structure, the way water flows across a surface, pooling gently around the stomata, those same pores on the underside of leaves that are actively searching for CO2 molecules to absorb. You are not just spraying your plant with CO2, you are flooding its immediate microclimate with a slow, dense, invisible river of exactly what it is trying to find, delivered precisely where the biological uptake happens. The efficiency of this passive delivery in a still air environment is genuinely comparable to active injection systems that cost hundreds of times more. Now, honesty requires a pause. This method has real limitations. A yeast reactor running on standard bread yeast and table sugar produces CO2 at a rate of approximately 100 to 200 millimeter per hour under optimal fermentation conditions, a figure documented in homebrewing literature and consistent with measurements taken by independent horticultural experimenters. That output is sufficient to meaningfully elevate CO2 concentration in a small, enclosed or semi-enclosed space of roughly 10 to 20 cubic feet around a single plant or small cluster. It will not transform a large open room, nor will it replace ventilation in a full-scale grow tent without multiple simultaneous reactors. If you are expecting industrial results in an industrial space from a $2 setup, you will be disappointed. What it will do reliably, consistently, and for almost no cost is rescue a plant that is CO2 starved in a confined corner, a tight balcony space, an indoor shelf, or any environment where still air is quietly strangling your plant's photosynthetic potential. For that specific problem, this $2 biological reactor is not a compromise. It is the most precise solution available at any price point. The only question left is how to build it, and that part is almost insultingly simple. You need 5 things: a 1-gallon plastic jug with a screw-on lid, a length of flexible plastic tubing, roughly 3 to 4 feet long, aquarium airline tubing, a small amount of waterproof sealant or silicone, 1 cup of plain white granulated sugar, and 1 standard 7-gram packet of active dry baker's yeast. The assembly takes under 10 minutes. Take your jug lid and make a single hole just wide enough for the tubing to pass through with slight resistance. Thread approximately 2 inches of tubing through the hole from the outside so the short end hangs inside the jug and the long end extends outwards. Seal around the tubing on both sides of the lid with your silicone, making absolutely certain there are no gaps where gas can escape. This seal is the only technical step that matters. If CO2 leaks from the lid rather than traveling through the tube, the entire system loses its directional delivery. Allow the sealant to cure fully before proceeding. Once the lid assembly is ready, pour 1 cup of sugar into the jug and add approximately 3 cups of warm water, ideally between 70 and 80 degrees Fahrenheit, which is the temperature range where baker's yeast operates at peak metabolic efficiency. Add your yeast packet, swirl the jug gently until the sugar begins dissolving, and then seal the lid firmly. Within 30 to 60 minutes, you will see the first bubbles moving through the tubing. That is CO2 produced live on demand for a total material investment of under $2. Position the jug at the same height as your plant or slightly above it, and direct the open end of the tubing so it hangs 2 to 3 inches above the top of the canopy, pointed downwards. Do not bury the tube in the foliage. You want the CO2 to spill across the leaf surface from above, letting gravity carry it down like a slow waterfall. The reactor will produce CO2 actively for 5 to 7 days before the sugar supply is exhausted. Total monthly cost to run one reactor continuously is approximately $1.80. Users report visible differences in new leaf development within 7 to 10 days, with measurable stem thickness increases in plants like tomatoes, peppers, and basil. You are no longer a passive consumer of a $9.7 billion industry. You are a grower who understands the invisible layer of agriculture they hoped you would never find. If this story has changed the way you look at the very air in your garden, please subscribe and like this video. We are dedicated to unearthing the history and science that they have tried to keep buried. Start growing your own future today. The next hidden treasure of the garden will be revealed very soon.